Welcome to the audio slide presentation of the work entitled Action Recognition via Bio-Inspired Features, the Richness of Centers Around Interactions. Action recognition is a challenging problem in computer vision, and many approaches have been developed. An interesting class of approaches relies on the extraction of spatiotemporal feature descriptors, and of course, one crucial question is to find the best spatiotemporal features to improve action recognition performance. The original contribution of this paper is to look at our visual system as a source of inspiration to define these features. So let's review a few ideas about our visual system. In the mammalian visual cortex, the information processing is split into different pathways depending on its nature, motion, or form. In both cases, input are processed in a hierarchical manner where each subsequent area integrates the information coming from lower areas. Motion processing is performed in the dorsal stream, where the most related visual areas are V1, MT, and MST. It has been shown that MT response correlates with perception, representing thus a crucial point for motion processing and a starting point for action recognition. So, what kinds of motion features does MT encode? MT neurons integrate V1 responses from the motion direction space inside the so-called receptive field. Most of MT neurons have centers around interactions where the surround modulates the information caught by the center, in most of the cases in an inhibitory manner. Different types of centers around interactions have been observed at the level of MT, mainly based on the shape of the inhibitory surround. Interestingly, the shape of the inhibitory surround is mostly asymmetric, making MT a good feature extractor of motion contrasts. Our goal is to evaluate how a richer motion representation given by the diversity of centers around configurations could provide an advantage for a high-level task such as action recognition. In order to achieve this goal, we propose a hierarchical model gathering basic facts of the dorsal stream, specifically V1 and MT visual areas, and thus obtain a motion representation based on bio-inspired features such as motion contrast. So how do we do that? We firstly consider that eyes are following motion, so the input videos were centered and cropped in order to have a desired spatial scale. The input video fits a V1 layer where V1 neurons, modeled as motion energy filters, are arranged in order to pave the entire visual field. At each position, several units of V1 neurons compute information for different motion directions and different space-time scales. V1 layer fits the next layer in the dorsal hierarchy, MT. MT neurons pull information from underlying V1 cells preferring a certain motion direction. Different populations of MT neurons are present, each of them having a different center surround interaction. Using the response of MT neurons as bio-inspired features, we built what we call a motion map representing the motion information contained in the input sequence. Each element of the motion map is the mean response of a certain type of MT neuron at a given location along the entire motion sequence. The quality of motion maps for action recognition was tested using two different databases, Baseman and KTH. Here we show three confusion matrices obtained for Baseman database for different configurations of motion contrast. Each confusion matrix shows a number of mismatch sequences. The inclusion of different MT surround geometries improved the general recognition rate from 63 up to 99% in the best case. Similarly, on the KTH database, the recognition rate was significantly improved with the inclusion of MT neurons with different surround geometries. Interestingly, we also evaluated the robustness of the system in three different cases, adding Gaussian noise, occlusion, and with a complex background. In all the three cases, the recognition was perfectly performed. In this paper, we have proposed a new set of motion-based features for action recognition. The contribution is that these features have been directly inspired by observation from the visual cortex, making this work an illustration of how it is possible to integrate biological vision insights into an artificial vision system. Following this line, we expect that progress in action recognition could benefit directly from progress in understanding brain mechanisms involved in this task. We hope you enjoyed this presentation, and we encourage you to read the paper for more details.